Did you know that researchers have genetically engineered bacteria that can detect TNT in buried landmines? Buried TNT and landmines are naked to the invisible eye, but obviously very deadly. So scientists came up with a really interesting idea. What if we could use bacteria as these sort of like glowing detectors to tell us if TNT is present in a field site? Well, that's what they did, and to pull this off, they had to tinker with bacterial gene regulation. So researchers stole genes from some marine microorganisms that glow in the dark and connected it to a regulatory switch that turns on in the presence of TNT. So these bacteria could be released into the fields, grow and divide, and in the presence of TNT, they will literally glow in the dark. So they've become a biological detector. This story captures just how powerful and practical gene regulation can be. Gene regulation can happen at different stages of expression. Whether a gene's DNA is transcribed into RNA, whether RNA is translated into protein, even regulation post-translation on the protein itself. But in bacteria, the main control point is transcription, deciding whether to even make an RNA in the first place. So let's start there. In bacteria, genes are often organized in operons. An operon is a cluster of genes controlled as a single unit, so the cell can turn a whole pathway on or off together. An operon includes a promoter, where RNA polymerase binds, an operator that acts like a switch, and the structural genes that code for proteins. So who flips the switch? Transcription factors. These proteins bind DNA and influence transcription. Repressors generally block transcription when bound to the operator, while activators help RNA polymerase grab hold and start transcription. So some operons are inducible, meaning that they are usually off until a signal molecule turns them on. One analogy that comes to mind for me is that I really like painting, but I'm not doing it all of the time. I have to have the right amount of time and energy and space to make it happen. And I'm not going to paint in the dark, so I like to imagine myself inducing or turning on a light, and if all of the conditions are right, then I can paint. But it's not something I'm usually doing as my default. Other operons are described as repressible. They're the opposite. They're usually on until some sort of product builds up to a threshold that the operon is turned off or downregulated. An analogy that I can think of is hunger. My baseline is I'm hungry, I'm always trying to snack on things, and I'm going to be hungry until I finally eat. And when I eat, it's going to signal to my body that I don't need to be hungry anymore. So for some time, that pathway is shut down because I've met my needs, but eventually I'll start to get hungry again until I finally eat something and it's repressed. A classic inducible system is the lactose operon, or lac operon for short. This example operon has been extensively studied in E. coli, and it allows these microbes to digest lactose. Without lactose, a repressor sits on the operator and blocks RNA polymerase from carrying out transcription. When lactose is present in the environment and is transported into the cell, it binds the repressor and makes it fall off. So the operon turns on, and the bacteria can make lactose-digesting enzymes. This makes sense. The cell wouldn't want to waste energy making enzymes to break down lactose if there isn't even any lactose in the environment to begin with. By contrast, the tryptophan operon, or TRP operon for short, is repressible. It makes enzymes to build tryptophan, an amino acid. When tryptophan is scarce, the operon is on because it's essential that the cell has tryptophan as building block for protein. But when tryptophan is plentiful, it binds to the repressor, activating the repressor. And so this repressor protein shuts down the operon. Again, this is saving energy. Why make pizza from scratch if delivery shows up at your door for free? Now, in this example, there's even a second layer of control called attenuation. This concept hinges on remembering that transcription and translation can happen simultaneously in the bacterial cell. So in my description, you'll hear me mention RNA polymerase, which is a key player in transcription, and also the ribosome, which is instead our powerhouse for translation. How does it work? The TRP operon mRNA has a leader sequence that can fold into different shapes. If tryptophan is abundant, ribosomes move quickly, and the mRNA folds into a stop signal that halts transcription. Again, this makes sense. If tryptophan is abundant, let's stop manufacturing it. However, if tryptophan is low, the ribosome stalls in its translational process, and this pause creates a different fold in the DNA sequence, allowing transcription to actually continue. Sometimes bacteria need to flip on many operons at once, like during heat shock or stress. They can do this with alternate sigma factors. Sigma factors guide RNA polymerase to promoters, so swapping sigma factors lets the cell redirect transcription to a whole new set of genes in one shot. Let's circle back to our TNT detecting bacteria. Scientists created this microbial sensor by pairing promoters responsive to a specific stimulus, in this case TNT byproducts, to a gene that encodes for green fluorescent proteins. Fusing the gene, which was stolen from other bioluminescent microbes, with a promoter that responds to TNT, creates a sort of switch that can glow in the presence of the explosive. So whether we're talking about bacteria glowing in the presence of TNT, digesting milk sugars, or something like adapting to thermal stress, bacteria rely on gene regulation. And for scientists, these regulatory pathways can be rewired or modified for a really wide range of applications, including environmental monitoring of our soils and waterways. And so there you have it. That's how we end up with bacteria that literally glow in the dark when they encounter hidden explosives.